She comes to us, another student from Gwinnett School of Mathematics, Science, and Technology. She's going to talk to us about gene expression cell regulators.
Yes, so I worked with Dr. Rinza Lee and Dr. Chunyang Lee in the third floor of this building. And we were basically looking at the role EAP2 plays in cells and in cancer. An introduction of EAP2. EAP2 is the ETS1 associated protein. It's a 362 amino acid protein also, and it has a molecular weight of 41 kilodaltons. We were looking mainly at its functions, and research has shown that it can be a negative regulator of ETS1. We have seen that ETS1 and EAP2 localizes in the mammalian cell nuclei, and it has also been shown to localize in cytoplasm. A big part of this project was on ETS1 and what it exactly is. It is a proto-oncoprotein, and it's part of the E26 family of transcription factors. And it plays a huge role in cell regulation, such as apoptosis and in cancer metastasis, invasiveness, and such. We looked at the interaction between EAP2 and ETS1, and it was shown that with higher levels of EAP2, as you can see here, they're going higher and higher, the MMP1 promoter activity went lower. So therefore, the transcription activity lowered with more. EAP2 has two forms. It has the long form and the short form. The long form has the N-terminal, that's the red part you'll see, and the short form is missing the N-terminal because it transcribes later on due to ATG mutants, which I will speak about later. And from the Western blot, you can see the long form and short form. So the purpose of over my 7 weeks here was to confirm that there are indeed two forms of EAP2, and that the short form comes from the ATG mutant. And also, we wanted to evaluate the role of EAP2 in promoter activity, and we used many tools to measure that. Also, we were looking at the different functions between long form and short form. During my time here, I did a lot of different experiments. I did cell culture and purification of plasmids, which dealt with the localization of EAP2. And I also did a little PCR. I used Illuminator, which measures promoter and luciferase activity, which I'll speak about later in an experiment, and especially Western blotting. So I'll just give you a quick overview of what a Western blot is. Basically, you start by getting the samples for testing. Gel electrophoresis is done by putting an electric field around the gel, and then the proteins travel. Transfer is when the proteins go from the gel to the membrane. Blocking, basically, it just blocks. Well, the membrane was chosen for its ability to bind to proteins. So blocking is done to stop the antibodies to bind to the membrane and to make sure that the Western blot results are clear and reduces what we call noise. Blotting and detection, that's basically when the primary and secondary antibodies are used tagged along, and the reporter enzyme, in this case we use luciferase, is used to, during the, when we go to the darkroom and expose the film, to actually see our results. And that's basically just a diagram of the steps. The first diagram is of the gel electrophoresis. That's a really good, actually, picture of what it looks like. And at the end, you can see the gel. And then on the other diagram, that's the transfer. So that's when the gel goes onto that membrane. So our first experiment was to find an antibody that can tell the difference between long form and short form. That is done by looking for the N-terminus of EAP2. We were also looking to prove the hypothesis that the short form is from secondary ATG. So in this experiment, we used beta-actin, the commercial antibody trial, and one we grew here at Emory, 2,4-D. Let's see. So for beta-actin, you can see that it's been loaded the same amount. That was the point of the beta-actin. Trial recognizes the N-terminus, which therefore would recognize the short form of EAP2. However, in this example, you can see right there, there's nothing in that spot, concluding that trial did not find the N-terminus. And 2,4-D, that recognizes, as we see here, this is the short form of EAP2, and this one right here is the long form of EAP2 due to ATG1 and ATG2 mutants. The results, therefore, are that 2,4-D showed that both mutants, they respectively generate the short form and the long form. And also that the molecular weight for each of the forms were quite similar. And the trial antibody did not recognize the short form, which further confirmed the hypothesis that EAP2 short form does come from ATG2M. And the other study we did, this was a functional study. We used lucifers. And a little background information. This is a diagram I just made. Basically, it shows that promoters regulate the protein expression. And in this case, it's the MMP1 promoter regulates the lucifers. And promoter activity is regulated by transcriptional factors. And in this one, we use the promoters CMYC, NMP1, and GM-CSF. And each of those transcriptional factors, SMAD3, ETS1, and NF-kappa B, they all regulate the promoter. A previous study has shown that EAP2 can actually regulate the activity of all of these transcriptional factors. So the results from this study, as you can see, the EAP2 level on all of these were quite high, indicating the promoter activity, while the ATG1 and ATG2 mutants on each one of them, they seem to be very similar, which was not what we expected. So because of, and this is the Western blot analysis right here, the MMP and the GM-CSF that we used. Right here you can see the grading proteins. So from these results, we could not really conclude, we could not find the functional difference just from this experiment. But this experiment did indeed confirm that there are the short form and long form of EAP2. And we'll still try to find the functions of both forms of EAP2. We need to keep looking at several cancer-related characteristics such as apoptosis and metastasis to try to find another way to find the difference between both of these forms and their functions in cancer. So in summary for my time here, and from the experiments done, we have seen, as I said, that EAP2 has the short and long form. It regulates promoter activity, which in turn regulates the protein. And we have not yet found the functional difference between the forms based on just promoter activity functional studies. These are just my literature. And I'd like to thank my mentors, Dr. Rinzal Lee and Dr. Chunyan Lee, for helping me around in the lab, showing me the techniques. Ms. Alyo and Dr. Lakowitz for being the program co-directors, and Ms. Monroe and Ms. Henry for communicating with us and keeping the program intact. And of course, Emory University and Bishop Cancer Institute for allowing me to do my research here. So yeah, thank you. Any questions? Okay.
So what was your favorite non-part of or other part of the program?
I really enjoyed the field trips. We took field trips to the Emory School of Medicine and to Grady Memorial Hospital. And I really enjoyed seeing the simulators and all the people there. They were very welcoming, and I learned a lot from those places. So.
What were they simulating?
Basically, they had for students who are going into, surgery and all that, they have simulators. They have, how should I say, plastic, corpses which move and talk to you and do all this crazy stuff. So, yeah, it was pretty interesting.
Other questions?